So, you're going to have your syllabus ready by end of second period. You are working on chapter 8. I will have that ready for you again this afternoon. The focus here is we're going to continue our discussion on cell transport and why it's important. So I'll just go ahead and dive into it. We talked about this idea that things move from high to low, and you were actually tested on it. This idea that because of kinetic theory, particles are constantly vibrating and moving around, bumping into each other. And if that's true, if you have a divider, if you have something that divides this material into two spaces, and that material is more on one side than it is on the other, then just by the fact that they're bumping into each other, they're going to move from one side to the other, one room to the other, if there's an opening for them. So that's the thought. And I just said that. I expect you guys know that, but let's be clear. I see some people staring into space, some people coming in late, some people don't have their notebooks open, they're kind of swishing their water around. They don't have their notebooks open even now after I've mentioned this 4 or 5 times. They're sighing, but they're not doing, right? And so this is a basic fact. You better know. I'm saying it clearly. That things move from high to low on their own because of average kinetic energy. And we measure average kinetic energy in the form of temperature. So because nothing is ever standing still, because nothing's an absolute zero, which would be the only way to get something to stand still, you're having movement, and that movement means that particles move from one side to the other, from high to low, without any added extra energy. Do they have energy? Well, of course they have energy, because if they didn't have any energy, they wouldn't move. Energy, by definition, is the stuff you need to move. Is that clear? And that takes a lot of different forms. It's confusing because it's ambiguous. There's not something you can point to, say that's energy. But it's the stuff of movement, all right? So kinetic energy is measured in the form, average kinetic energy is measured in the form of temperature. Now, that being said, we have these membranes, cellular membranes, or what we call another word for them, again, vocabulary words, the plasma membrane, and I have to write with my finger until the pencil's charged. The plasma membrane is selectively permeable. And you just got tested on that idea of selective permeability. That means that if you have a cell, if there's a cell, and it is in a solution, and in the case of a beaker, in this case, and obviously it's a gigantic cell, there is a difference in concentration of particles, then there's a question on whether stuff can get through or not. There's a question, can material get through the membrane or not? That question is a tough one because it depends on this cell's makeup. Because the cell, as you know, the cell membrane isn't just a bunch of phospholipids. If it was just a bunch of phospholipids, you could pretty easily tell what was going to get through. Small polar molecules, correct? Small polar molecules can get through a phospholipid bilayer. Nonpolar materials, small or medium-sized nonpolar materials can get through pretty easily because these molecules, these phospholipids, are both polar and nonpolar. They have the polar heads and nonpolar tails. We've studied those. We know that. But we also have these structures that we've talked about in class numerous times that I hope that you have straight in your head because you're going to need it. You have these things called proteins. And these proteins sit in the membrane, and they change the permeability of the membrane. Permeability is the ability for substances to get through. So this is a basic concept here, this vocabulary word. You could even think of it as an SAT vocabulary word if you like. It's permeability means the ability for materials to pass through. So if something's permeable, that means it's able to get through the divider. If your shoes are impermeable to water, what are they? Huh? Waterproof. Because the im means not. Impermeable means not permeable. But selectively permeable means what? What does selectively permeable mean? Selectively permeable. What does it mean? A charming smile is not sufficient. You must go ahead and tell me. Yes, I'm asking you. Go ahead. What does selectively permeable mean? Say what? It's not permeable to all things. It's permeable to what? To select things. That's correct. It's all in the title. It's not difficult. So what determines what gets in and out of the cell? Well, the basic concept of nonpolar, of course, small, polar, those are pretty permeable. So water will get through, usually, all times in biological molecules and biological systems. Small, non-permeable, polar. But the question of whether ions get through or not, the things that don't get through easily, ions, large, and I think that large polar was on your test. Oh no, it was on the AP test. Large polar molecules. They have a harder time getting in. They need assistance. So when things move across a membrane, when things move freely across a membrane, like water, for instance, moving in both directions, remember that these materials move in both directions. This is random movement. So you really don't, it's not like somebody's choosing. There's no choice here. They just randomly move. We take our hoods off our heads. Two people have their hoods on their heads. Could take them off your heads. Thank you. So water moves in and out of the cell pretty easily in both directions. The question is, in which direction does it happen to move more? So that's permeable. The cells are generally permeable to water because they're small and they're polar. The next question is, what about ions? What different kinds of motion would there be? So before we get into that, let's talk about the different kinds of solutions that a cell could be found in. So you might find a kidney cell, a brain cell, and an intestinal cell, or a blood cell. Or you might put this material in these cells in three different solutions. When we're talking about the concentration of solutes, that's what we're talking about. The concentration, everything we've been talking about all first quarter, the concentration of solute. We're going to call that tonic. It's a suffix tonic. How much solute you have, we're going to call it tonic. So, what are the three different? This is going to fall out of your pocket, or it's going to break. Putting it in your back pocket is the worst place to put a phone. Tonic. What are the three possible situations you can have when you're talking about a tonic solution? Three possible situations. Three possible concentration differences. Yeah. Just give me one.
When there's more solute outside of the cell than inside?
So there's more solute outside the cell. I'm drawing it here. Then you have inside. Do you agree that there's a higher concentration of solute outside in this example than there is inside the cell? Does everybody agree with that? The solvent is what? On both sides, the solvent is what? In a living system, now I see your hand up, but I want someone else to answer. In a living system, what the solute is whatever we're talking about, it can change. What is the solvent always in a living system? I'll wait, because if you don't know this, we have. Just have to sit down and let you read a book or something. Yeah. Water. So water. Every one of you should have immediately said water. If there's a question in your mind on what the solvent is for a living system, you need to review, reflect, and revise your thinking. It should be water immediately in every living system on planet Earth. Period. End of story. All right, so water is a solvent. Do you see why I spend a whole week or two on water?
Yes.
I hope you do, because I don't understand why anyone even paused with that answer. All right, so higher concentration. What's the concentration called? It's concentration of solute is called what? Tonic. So we know it's going to be some kind of tonic. Everybody agree with that? Some type of tonic. It's higher outside than inside. Relative. If I ask you, what is the concentration of this solution? It's some kind of tonic. What would it be? Does anybody know? Just before I say it. Yeah. Hyper is correct. This is hyper. Hyper because hyper means high. When somebody's hyperactive, they're highly active. When somebody's hypoactive, they're not active. So hyper means high.
So high solute?
High solute. High solute. Now this is a danger. This is really easy. Let me make this very clear. If you hear my words and you process this information, it is the easiest thing you have to do in biology, if not the second easiest, or whatever. It's one of the easiest things you can do in biology. Deciding if something's hypertonic, hypotonic, which way is water moving. That's simple. If you get the idea straight the first time. The danger here is that it's very easy to confuse it just by not paying attention. I can tell you there's about 10 people already this year that confuse right from left. You know these people, right? Yes? Some of you may be these people. Yes or no? Yeah, okay. Just right and left. Easy to do if you practice it and you got it straight. It's not easy if you confuse it, right?
Yeah.
So we got to make sure we get these definitions straight. Hypertonic means higher outside. We'll get back to why that's important. What's another situation? What's another possible situation a cell could find itself in? Yeah.
Hypotonic solution,
Hypotonic, what is that?
where the solvent is concentrated higher inside the cell than outside.
Solute.
Oh, sorry, my bad.
Yeah.
Solute.
So here in a hypotonic, you have less outside, because we're talking about outside. If we say the solution, when we say the solution, we generally mean, here's another, well, we'll have to come back to that because that's going to be somewhat confusing as well. So this is hypotonic. So hypotonic, again, is lower outside than inside. Lower what? Lower solute. Higher solute, lower solute. So what's the one that we're missing? Which is the third possibility. It's either higher, lower, or equal. And that we call iso. Iso means the same. It's the Greeks. They did it to us. So iso means the same. The same amount of solute in divided by, remember we're talking about solute divided by solvent. Amount of solute divided by the amount of solvent, usually in volumes or grams per milliliter or something like that. All right? Could be percentages. You could eat a lot of problems. It could actually be a decimal. It could be a fraction. It could be a decimal, or they might give you a percentage. Right. So there's two possible ways they can give you that information. A decimal point something, 0.002. They could be a percent grams per milliliter. It could be a fraction, or it could be, I hate writing with my finger. It could be a fraction, it could be a percentage, or it could be a decimal. But whatever, it's either going to be bigger or smaller. Does that make sense? It's either going to be bigger or smaller. Either the percentage is bigger or smaller, or the decimal is bigger or smaller, or the fraction is bigger or smaller, whatever, when we're talking about whether something is iso or hypotonic. It's just that simple as far as that's concerned. We all have this straight in our heads? No questions? Yeah.
How would you determine by, let's go for example, how would you determine by percentage if a solution is iso or hypotonic?
All right, let me give you an example. So if you have a cell and it's 10% solute, 10% sodium chloride inside and it's 20% outside, what is this?
Well, that would be hypertonic.
Hyper, right? If I drew it again, see how easy this is? I love these. These are real easy to make tests for. 50%, and outside it's 20%. It's hypo, and if the solution is 0.2 and outside is 0.2 grams per milliliter, it's iso. So you can have it any which way. You're just comparing inside to outside, inside to outside. One side, one room to another room. One cell to another, one space to another space. Concentration of solute. Are we clear? That's all there is to it. There really is not much more to it than that. But then There are some, of course, like everything in life, everything's based on simple. Do you guys not notice when you spill hot chocolate? When you walk, just out of curiosity, when you guys walk along and you spill hot chocolate on the ground, do you all not notice it? Do you think the kids just don't notice it? They just leave it there, or do they notice and just keep walking? They just notice and just say, "Yeah, I don't care. It's not my problem." All right. When somebody slips in hot chocolate, that's just going to be awful. Yeah? I can't hear you. Yeah, sit wherever you want. All right, so moving on. So that's hyper, iso, and hypotonic. Seems simple. Now, so if I give you a situation where either I do this, right, where you got one side is 10%, and I tell you it's 10% oxygen gas, and it's 20% oxygen gas. This could be happening in a blood cell, in the lung. I mean, it could be happening everywhere. We don't know. Quick question, what's going to happen? This is a selectively permeable cell membrane. So let's call it a cell membrane. What's going to happen? You should be able to tell me what's going to happen. Let someone else. Let someone else. Yeah.
I think the higher one is going to try to go lower.
Is it going to try or is it going to do?
It's going to do.
Huh?
It's going to do.
It's going to do, yeah. It's going to go, the higher, the 20% oxygen is going to move. Oxygen is going to move in both directions. It's going to move more in one direction than the other. It's going to move more from the 20% to the 10%. Is that clear? Yes? All right, so it's going to move in that direction. Why? Because oxygen gas is nonpolar and it's small and it passes through the membrane easily. What do we call that kind of movement? From high to low with no, there's a word for it. Yeah.
Diffusion?
Diffusion's correct. So this is called diffusion.
Diffusion from high to low?
Yeah. High to low, random movement, mostly in one direction. Diffusion is happening in both directions. Notice it's going in both directions, but the movement is mostly into the left. Is that clear? All right. So we call that diffusion. Now, what if I have this same cell membrane, 10% and 20%, but instead of oxygen, it's sucrose. Sucrose can't get through easily. It's still a cell membrane. Still a cell membrane. 20% sucrose. It can't get through, right? Because I'm not telling you there's any special transporter. It's not getting through. I'm telling you it's impermeable. In the problem set, whenever you get a problem like this, I'm telling you right now, any problem you get in this class or on any state exam, they will have to tell you in one way or another if the substance crosses the membrane. Because anything could cross a membrane under the right circumstances, if you have the right transporter, if whatever. So you have to be clear. Now, I want to make sure you understand, they will give you clues as to whether it passes the membrane without necessarily telling you. Just telling you it's sucrose, they expect that you know it doesn't pass the membrane without help. And if they don't tell you there's a transporter, you know it doesn't pass the membrane. Does that make sense?
Yeah.
That's the level they need you to understand. Yeah.
So, would you just, if they say, which way would it go, would you just say it's impermeable?
It doesn't go anywhere. Sucrose won't move, but they won't ask you that. What they're going to ask you is what happens.
Oh, okay.
Something will happen. It's just sucrose won't move. And that's the next part of this discussion. It gets a little complicated, not hard, again, if you're following along. So sucrose, when you have a membrane, cell membrane, that's not permeable to, so let's go ahead and draw this out. So again, I keep warning you, you guys should draw these things out so that you can visualize it. So if you have on one side 20% sucrose, and on the other side it's 5% sucrose. Now the water, the solute won't move, it can't move. If the solute does not cross the membrane, does not move across the membrane, okay? If the solute does not move across the membrane, in this case sucrose is polar and big, so it can't cross the membrane. Nature, there you have a higher concentration, it's more crowded out here than it is in here. So what do you think happens? Yeah. That's right. The water will move in. The water will move. Why? Because it's crowded. Think about a really crowded gymnasium. Think about the gym. It's hot. It's what's the dance you guys do? A homecoming dance. You all bought tickets. They oversold the tickets. Everybody's like elbow to elbow, face to butt to face in that place. Like it's hot and steamy, and somebody opens a door. What happens? People go out that door, right? Does that make sense? So if it's possible, if you're overcrowded, outside it's overcrowded, the solute can't move, so this is not moving, sucrose is not going. Then what does happen is water moves. Water moves. Now, interestingly, water is not going to move like you would think, well, water's moving, so it's going to go from high to low. It's 20% down. No, it's the opposite. Where water's concentration is the highest, it's going to move to where water's concentration is the lowest. This is a tricky part. This is where it seems backwards. Yeah.
Is this osmotic pressure?
Yeah, this is going to be osmosis. It will be osmosis as soon as we define it. So water, when we talk about percentages, what do you think about when you think about a percent? If I say 5%, what do you think about? All right, out of 100 is good. What do you think about from middle school, from elementary school? Somebody said percent. What did they use to draw it? A graph? What kind of graph? A pie graph. Everybody always uses a pie graph because pi represents 100, right? So we can do the pi is 100%. A pi is 100%, okay? What's 5% of that 100%? What would 5% be? Like some kind of sliver, right? Agreed? So this is your 5%. Well, wait a minute. If that's 5%, if this pi, this represents your sucrose, what's the rest of this? Water. That's right. Again, what is the solvent of life? Water. So H2O is 95% in here, right? And 95% water is 5% solute. Does everybody see that? I hear some people saying, some people are staring. Some people are busy writing, that's good. But do you understand? Let me hear you say, or I know if you're on your phone, give me your phone. Give me your phone. Come on. Just hand it to me. Yeah. No, it's just an asterisk. I'm sorry. If anybody else needs help focusing themselves, thank you. Let me know. So you have 5% solute, 95% water inside the cell. Now, what about outside if we did the same thing with on the outside? How big is that sucrose wedge? Bigger or smaller than 5%? It's bigger. Probably drawing this horribly, but we'll go ahead and just assume it's right. And this is going to be, this is 20%. So if it's 20% sucrose, what's the rest? 80% water. Yeah.
So like to get this right, on the inside it's like 5 and 95, and on the outside it's 20 and 80.
That's right. But what people forget, what people don't get in their head, what people miss all the time is that 20% solute means 80% water.
So it's basically 100 minus the solute rate.
It's 100 minus the solute. That's all it is. So where is water the highest? Where's water? I'm using two colors on purpose. Is that clear? So if we're looking at a hypertonic solution, the solute is higher outside and lower inside, correct? But if we're looking at water, water is what? Higher inside and lower outside. So water is higher inside and lower outside, where the solute is higher outside and lower inside. They're going to be opposite each other. Does that make sense? All right. So if the solute can't cross, water will cross. But if it's going to be passive, what do we know about passive transport? What must happen? It must go from what to what? High to low. It has to move from high to low. So it has to go from its high to its low. Water has to go out. Water has to go out. It's the only way it's going to do it without being forced in. So water goes out to fill in the spaces. And so what happens is outside becomes more dilute and it gets closer to equilibrium. So you'll reach some kind of equilibrium on both sides of the membrane. Is that clear?
Yes.
All right. A dynamic equilibrium. So somewhere in between 5 and 20%, something like 10, 15%, 10%, whatever it is. You can do the calculations. The bottom line is that somewhere in between 5 and 10% or 20%, you'll get a dynamic equilibrium where there's movement of water going both directions equally. But in the beginning, most of the water is going out. Some's moving in, but most of it's going out. The majority is going out. Yeah.
So water won't move to the lower solute concentration, it'll move to the lower water.
Higher solute concentration. I'll get to that in just a second. I'm going to summarize all this in just a second, but I need you to try to understand it so that you don't get confused, because I'm telling you, this is where everybody gets confused and they get wrong answers, and it's sad because it's really easy points. So high to low, 20%, 5%, that's what we're talking about when we're talking about this. By the way, this movement of water, the diffusion of water, is also known as osmosis. All right, so you should know that was tested. But osmosis is the movement, is just the diffusion of water. Water moving from its high to its low. When does water move? When does water move this way? Yeah.
When the solute's
When the solute cannot move. Water moves in order to balance out that concentration. It's all about finding equilibrium. All right. Let's summarize this. So there's a general 2 rules of thumb. There's a couple ways you can think of it. You can think of it, it doesn't matter to me how you think of it. All the ways I'm about to describe to you work. So whatever makes sense to you, you go with it. So the first thing is, if it can move, rule number 1, if it can, the solute will move from high to low. That's it. If it can, the high-low concentration. I should put close the door, will you? So if it can, the solute is going to move from its high to its low. Is that clear? So that if it's oxygen gas, it's going to go from its high to its low. Easy. If it's whatever it is, it's going to move from its high to its low. Rule number 2, if it can't, I shouldn't put it because it's going to confuse you. If the solute cannot move, then water will. So water will move if the solute cannot. Thank you. If the water will move if the solute cannot. And that has to be clear in your mind. Now the question is, in which direction will water move? So there's, again, several ways you can think of it. But the easiest way to think of it, in my opinion, is water will move toward the higher solute concentration. So the higher solute concentration. So water is going to move towards wherever it is hypertonic, wherever it's hypertonic. Yeah. Yeah. Water will move towards this higher solute concentration. There's another way to think of it is, let's just call this A. Maybe this doesn't work as well for you as B. B is water will move down its So water, the other way to think of it is water's going to move from its high to its low. Either way works, but yeah.
Is that why hypertonic, like cells in a hypertonic solution may shrink up and lose mass?
That's exactly right. We're going to get to that in just a second. That's the point. We'll have to do it Wednesday because remember tomorrow we have SATs. You guys are leaving early. If you didn't know that, read your bulletin on Sundays or Monday mornings before school so you know what the heck is happening. So if you would have read it last night, you would have been able to tell Ma, Tuesday I'm leaving early because we got PSATs. Not according to your face, you didn't. That's what you got, PSATs. Whatever. You know you were shocked because you didn't even know. Just admit it. Just admit it. Admit it. Admit it, Parker. Don't shoot a spider web at me. Come on, that's funny. That was good. How could you say what you're saying is corny? Oh, I hate when you kids say it's corny. Water will move down its concentration gradient from its high to its low. Everybody get it? All right, so let's go ahead and take a look at why that's important. Let's take a look at a plant. Let's take a look at a plant. A plant grows up, and anybody makes fun of my plant, you're going to be up here drawing, and you're going to be a limited amount of time, and you're going to do it with your finger, and you're going to tell, and then we'll talk about. This looks ugly. All right, keep talking. You're going to be up here, and I'm going to be laughing at you. I hear you talking. It looks like what? Go ahead, say it. Go ahead, say it. Go ahead. It looks like what? So we've been growing it. Look. We're not gonna. I'm drawing with my finger. I got minutes. You guys are gonna start getting up and moving out in a minute. Just chill. You know. You've grown some plants from seeds in this classroom, and you're growing them from seeds in this classroom, and there's going to be flowers coming up soon, I hope. We're going to replant them. And in any case, you have water you see that we're watering here. Why does water go into the roots, up the stems? Yes, we know it's hydrogen bonding. Up the stems and into the leaves. Oops. Why does water do that? And do you ever ask yourself, how does a plant go straight up? You ever ask, look at these things. They're popping straight up. You're growing cells. Why doesn't it just like flop into a ball? Why do they, besides cell walls, you have cell walls, right? So you have these little empty cell walls. They're little building blocks. Plants have cell walls. They're not really rectangles, perfect rectangles, but let's just call them that. Let's just draw them like that. And they start building on each other, and they start growing upwards. If you took a picture of a microscopic slide of them, inside of the cell walls is what? Well, a cell membrane. You're correct. There are organelles. So inside a cell membrane, there's a cell membrane here. What's inside a cell membrane? What's all around the cellulose wall? The membrane's made of phospholipids, correct. What's all this in? What is the solution of life? Water. So do you ever wonder why a plant that's dying is droopy? It's low on water, is correct. So what happens is instead of going upright, a plant might do this. So, you've seen this happen to plants before? All right, they droop. That drooping is happening. It's happening because they're low on water. Well, what makes them stand up straight then? Well, water. But how does water do that? Because water goes to where there's a higher concentration of solute, and then it pushes out, right? It pushes outwards against the cell walls. That's what holds these blocks up. That's what holds these blocks up, is water pushing out. There's a force of water. It's like a water balloon. It's like a water balloon. A little stiffer than a water balloon, obviously. But if you want to give the thing some shape, some resistance, you have to have it filled with water. Is that correct? Is that understood? Otherwise, it starts to droop. The cellulose can't hold itself up. Yeah.
Could you repeat like that in like the actual like word?
Huh?
Could you repeat that?
Huh?
Could you repeat it?
Repeat what?
What you just said, how plants stand up. It's water.
Water pressure. Water pushes on the inside of the cell walls. Water pushes on the inside of the cell walls, and when it does that, the plants stand up. They grow upright. They start moving. All the blocks line up, and there's a force that holds it up. Otherwise, gravity would just pull it back down. Is that clear? All right, we call that Tuger pressure. So Tuger pressure is what pushes the plant up. Yeah, can I finish my thought though? Thank you. So if you put a plant, let's say the plant there's a road. Let's say there's a road, and you have plants growing along the side of the road, maybe a tree or something. What do we do in our roads in Cleveland? I don't know why people aren't writing this down. What do we do in our roads in Cleveland in the winter? But we drive. Oh, that's so nice, because no one ever drives anywhere else in the world on the roads. It's just Cleveland's the only place we drive on the roads. All right, what do we do in Cleveland that you don't do in Florida or in Arabia? You put salt on because of the ice, right? And so we put salt on the side of the roads. So now what is this solution of water that's going into the ground here where the trees are living? Salt solution. Is it hypertonic, hypotonic, or isotonic? Hypertonic. So where is, I like how people put stuff away. It's not, class is over. Hypertonic. Where's the water moving that? Yeah. Higher. So in which direction would water move in this case? Which is where? In the ground. So the water will move down. So you're just killing. That's why plants don't grow well where there's freshwater plants don't grow well. There are saltwater plants, right? But freshwater plants don't grow well in where there's a lot of salt. Because what happens is that the plants, by the way, you just did an experiment where you put salt in different plants. And I think for the most part you saw, when done correctly, the salt water killed the plants. Why? Because the water went where? Into the ground. The water left the plant. You dried out the plant. You dried out the plant. That's why we salt our fish. That's why we salt meat. Why in the old days did they salt meat and salt fish? What did that do? It dried it. It took the salt out of the meat. And that made it last longer.